Live cell imaging is the study of dynamic processes in living cells. When using high magnifications, a consumable with a thin bottom material is required. It starts with the seeding of the cells. With the Eppendorf Cell Imaging Dish, the cells can easily be concentrated on the central imaging area. Just pipette the cell suspension directly on the glass bottom. This central imaging area with the cover glass bottom is a specifically designed cavity. It is located 2 mm below the surrounding plastic bottom. Thus, the liquid does not splash over, and the cells are kept on the glass bottom. After seeding the cells, put them back into the incubator and give them time to attach. The glass bottom of the Eppendorf Cell Imaging Dish is tissue culture treated to support cell attachment of adherent cells. The time it takes for the cells to attach to the surface depends on the cell type you are working with. Make sure to check with the microscope before you proceed with your experiment. Live cell imaging is the balance between getting the cellular information needed and keeping the cells alive. Labeling is not always necessary when studying living cells. Many cellular processes can be visualized directly on the microscope using phase contrast techniques. When staining living cells, the use of pre-warmed solutions avoids temperature shocks. For long-term experiments, fill the complete dish with the required medium to reduce evaporation. Cells will only behave naturally when they are incubated at conditions that mimic their natural environment. Especially during long-term imaging experiments, it is crucial to maintain these conditions to achieve physiologically relevant results. For live cell fluorescence imaging, it is important to reduce photobleaching by limiting exposure time and light intensity as much as possible.